start recording to this lecture for us. And again, if you have any questions or anything as we're going along, please don't hesitate to unmute yourself and speak up or post it in the chat. I will try to keep up with what we have there. It's the 1st of March. Yay, we're getting closer to spring every day. Like I said, this is our topic on carbohydrates. So we're going to talk about carbohydrates really for the next 2 weeks. So this week, we're going to talk about how they're made. And then next week, we're going to talk about how we can extract the energy out of them. So lots to do with carbs. This week is kind of structured very similar to last week, where we're going to have a lecture on Monday and Wednesday, and then we'll do an IRAT, TRAT on Friday, or some activity with this. I'll kind of get a sense on Wednesday if we'll do a quiz, or if I think we should spend more time maybe working on the material together. But it will be a team-based activity. So we will do a team learning thing. Right now it's scheduled for an IRAT, TRAT, but I'll let you know Wednesday. We'll get a sense after class what we're going to do with that. And then there are 2 homeworks for this week. So there is one on carbohydrates, it's relatively short, and one on photosynthesis. Again, both of those are due by Friday. And then we have our quiz 4 on the vocabulary from chapter 10. So that'll be due on Sunday. All right. Oh, goodness. I forgot to change the title. This should say carbohydrates, not reactions and enzymes. But we do have the right learning objectives here. So we're going to look at the structures of carbohydrates today, in both their ring forms and in some of their linear forms, and how just changing minor things on them can really impact how that carbohydrate is able to be utilized in a particular organism. So really cool there. We'll talk a little bit about some of the nomenclature, how we name these different sugars, a little bit similar to what we saw with our proteins. So hopefully some of these prefixes are looking a bit familiar at this point. And like I said, we're going to look at the consequences of what happens when our carbohydrates form in linear strands versus some very branched strands, and how that's going to impact how they function. So really cool structure-function relationship still. So I'm going to try and get through as much of this as I can today, but this is the roadmap from our book. So just real quick, what are carbohydrates? Carbohydrates are sugars. All right. So any carbohydrate that we're talking about is, a type of sugar. These are one of our large macromolecules that gets assembled through dehydration synthesis or polymerization reactions. Very, very important for the energy source of most living organisms. So that's one of their really important functions. They're also going to contribute to the structural properties of many organisms. So we're going to see how they help supply structure to the cells. And then sometimes those cells' main function might be to provide structure as well. Carbohydrates are also interesting because they are involved with our immune system being able to recognize and identify cells in our body as belonging to us. So carbohydrates can be used for identification. So another neat feature of them. And then I just broke down here some of the prefixes that you might see associated with some of our sugars. So there are different terms that you're going to see as we kind of walk through the next 2 weeks that refer to sugar. The term saccharide is a very general term, right, that refers to a sugar, and it's not specific to glucose, where we're going to see some of the other terms we use tend to be in reference to glucose more than just a general sugar. So a saccharide is a general sugar, and I'm sure some of you have heard of the artificial sweetener saccharin. They got their name, right, from taking it from saccharide. Mono just means one. So we're going to start with looking at monomers. And then we're going to look at some larger structures. So similar to what we saw with proteins, we have oligo, which is just a smaller sugar compared to polysaccharides, which are where we're going to spend most of our time. So looking at monosaccharides and polysaccharides. All right. This table is actually from a different textbook, but I thought it was really important and I wanted to share it with you all because it kind of broke things down in a really simple way. So this is a really good slide to use when you're kind of studying and trying to get a grasp on what all carbohydrates do. So over here on the left, we see that carbohydrates are made up of carbons, hydrogens, and oxygens in a very specific ratio. And we'll look at this ratio here in just a minute. But basically, for every carbon, we're going to see 2 hydrogens and 1 oxygen that make up the actual structure of the carbohydrate. The little n here tells us how many times this pattern is repeated for a particular carbohydrate. So we're going to take a look at that, but it's very well structured. It's, carbohydrates are pretty simple in being able to recognize them because they have this pattern. The monosaccharide will serve as our basic building block. So as we're building our polymers, we're basically just joining monosaccharides together. You can see over here, I don't know, it's not giving me my, there we go, my pen today. All right, so we can see over here, glucose and fructose are 2 very important examples of these monosaccharides. So just a simple sugar. Sucrose, which is your basic table sugar, the sugar you use when baking or things like that, is a disaccharide. So we've linked 2 monosaccharides together. And then we get to the really fun ones, which are these polysaccharides, such as starch and glycogen, which we can see here. These sugars are really good for long-term energy storage. All right. So starch is found in plants, and animals will store their sugar as glycogen. And then down here, we have a couple examples of structural carbohydrates in the form of cellulose and chitin. And so we're going to compare these structures today so you can see why starch and glycogen can be used for energy, whereas cellulose and chitin, although very similar, cannot be used to store energy and therefore are used to provide some structure to the organisms. So here's that little formula that I talked about on that last slide. So this is probably a good thing to highlight in your notes so that you can recognize when you see a sugar. All right, so again, we have 2 hydrogen, basically a molecule of water, right? For every molecule of carbon we have is one way you can remember that. And it makes sense, right? Because carbo refers to the carbon and hydrate refers to the water. So you'll have a carbon and a water basically to help, that builds these carbohydrates. So a name that actually makes sense, right? And so the N then is how many times I'm going to repeat that pattern to form a particular carbohydrate. And sometimes it'll be small, maybe only 3 of these, up to thousands of them. So the range of sizes of carbohydrates is quite extreme. What we're going to see is that even though we do have a lot of carbon and hydrogen bonds, which is typically hydrophobic, right? So these have pretty similar electronegativities and like to share their electrons. The fact that we have these oxygens in play is going to actually result in carbohydrates being polar. So we do see that we will have some of these carbonyl groups, so carbons double bonded to oxygens, as well as some hydroxyl groups. So these are what are going to allow the carbohydrates to interact nicely with water, but it does have a significant number of carbon-hydrogen bonds as well. So just something to keep in mind. Overall, we will consider a carbohydrate, though, to be hydrophilic. And then this is just a picture. You don't have to memorize this, but I wanted to show it to you in comparison to when we looked at a salt, so sodium chloride, that was interacting with water. So sometimes the term, that something dissolves in water, it actually has a couple different, meanings. So here, the black and white kind of structures you see here are your carbons, are the black structures. The white ones here are the hydrogens. I'll just kind of label a couple here. And then these red ones are oxygen. Okay, so within here, we have this nice little sugar molecule. Unlike salt, when we put that in water, the sodium and chloride ions, remember, kind of that those bonds, that ionic bond kind of breaks, and they really mix around with the water, and the individual ions will get surrounded by water molecules. And we'll say that, a salt dissolves in water. Well, when we look at sugar, we often say that sugar dissolves in water too, but the big difference is that the actual sugar molecule itself isn't really breaking apart. So the sugar molecule here is held together by covalent bonds, and so they're really strong. And so the water molecules, rather, are going to just form hydrogen bonds, as you can see, these little red dots, around the sugar molecule and kind of surround the whole thing. So it looks like it's masked in water, but the actual sugar molecule itself is still intact. Does that make sense to everybody? So although it dissolves in the water, the actual molecule itself isn't breaking apart. It's just kind of getting masked in the molecules of water. So a little bit different than what we see with salts and ionic structures. All right. So what we're going to look at now is hopefully helping you be able to distinguish one monosaccharide from another. So if you're really good at like those hidden picture puzzles or finding what's different between two pictures, this topic today is the one for you. So it's a little bit of a scavenger hunt. So what we're going to see is that monosaccharides are going to vary in their structure in four main ways. Okay, so the one thing that we can look at is for that carbonyl group. So remember, carbonyl is the carbon double bonded to our oxygen. All right, so we're going to look at where that's placed within our carbohydrates. We're going to see that if this carbon double bonded to oxygen is placed at one of the ends of our molecule when it's in its linear form, it's going to be referred to as an aldose sugar. Where if we find the carbonyl group located in the middle somewhere, we're going to refer to it as a ketose sugar. All right, so we're going to look for that on the next slide, or in probably 2 slides. The number of carbon atoms present. So how big is our carbohydrate, right? So does it have 3, 5, 6, even bigger? We're going to try to stay around a hexose carbohydrate with our talks today for the most part. So again, we're looking at, you can use the prefix to kind of give you the number of carbons that are going to be present. And I hope you're starting to see a pattern with the endings of these words. Does anybody notice that there were a lot of OSEs that showed up on this slide? Does anybody know what that refers to or have a guess? Yeah, Christoph, this means sugar. Perfect. Yeah, so anytime you see a word in this class that ends in OSE, you know that's going to be a sugar. All right, so that'll give you some indication of what it is. All right, so here is our aldose versus our ketose type sugar. I'm going to close the chat for just a second because it's blocking my screen. But what we can see here, this is a linear form. This is a really small sugar, right? So they've only given us 3 carbons. And so they've numbered the carbons with carbon 1 being at the top and carbon 3 at the bottom. And they've highlighted for us that nice carbonyl group. So we see here that this would be our aldose sugar because it has the carbonyl group located at the end of the chain. Where over here in the right, we have our carbonyl group located here in the middle. So how can you remember the difference between an aldose and a ketose? So I simply just use the alphabet. So A is at the beginning of the alphabet. So if my carbonyl group is at the end, the beginning or the end of my chain, I know that that's an aldose, A right at the beginning. A ketose, so K's are kind of towards the middle of the alphabet, right? So if my carbonyl group shows up somewhere in the middle of my linear strand of my carbohydrate, I know it's a ketose sugar. So that's how I remember the difference between an aldose and a ketose. Does anybody else have another way that they remember the difference? Maybe those of you who have had chemistry. All right, so we got some ideas coming in the chat. Nope, somebody had something else going on. All right. All right, so you guys think you can handle that one? I know you guys are really quiet today. All right, so now remember there were 4 big things that we had to look for to tell our monomers apart. So we've got, where the carbonyl group is, we have how many sugars are present. Now we're going to look at the arrangement of hydroxyl groups, okay, within our single monomers here, all right, so our monosaccharides. So if we look at them, they're kind of crazy, right? They're like all over the place. So we're going to take a look. Let's focus here on glucose and galactose for a second. All right, so I'll give you guys a little brain activity. Who can tell me where the difference is between glucose and galactose? And we're going to start by numbering this as carbon 1. So our carbons are located, remember, at the points, and carbon 2, carbon 3, 4, 5, and 6. All right, so whoops, I went the wrong way. I was trying to scroll up the chat. All right, so I'm seeing where the bottom OH is connected. Which carbon? Carbon 5, thanks Jackson. Yeah, so carbon 5 looks a little different, right? So if we look at carbon 5 on galactose, we see that hydroxyl group is pointing in one direction, and over here on glucose, it's pointing in another direction. Yes, these are isomers. Yes, Jackson, you're absolutely correct. And so that's the only difference, right, between glucose and galactose. If you look at these structures, right, is the direction that this hydroxyl group is pointing. This has a huge impact on whether or not we can break the sugar down. So if you were to feed, say, yeast, because they like to eat sugar, galactose instead of glucose, you're going to see a significant decrease in the amount of sugar that they can break down and the amount of carbon dioxide that they give off, because they can't break this down as well as they can glucose. So just that little change is going to have a huge impact in function. We're also going to look at the differences between their linear and ring forms. So typically we're going to see that our sugars are going to want to go into their ring structures when put into an aqueous solution. This is going to allow them to be in their lowest energy conformation. All right, so here is glucose and galactose shown in their linear forms. Okay, are these maltose or ketose sugars? We'll quiz you on that right now.
Ketose.
I see one. Okay. I'm seeing ketose in the chat. What makes you think it's ketose?
Because they're not on the end.
So remember, for determining if it's an aldose or a ketose sugar, we're looking for a carbonyl group. So we're looking to see where carbon is double bonded to oxygen. So if we look through our structure and we look at carbon 1 here, we can see we've got that carboxyl group here. I should have circled the hydrogen. And we have a carboxyl group here. So these would both be aldose sugars. Does everybody see that now? So that wasn't what the picture here is highlighting, right? This is actually showing us hydroxyl groups. But I wanted us to see, could we figure out if they were aldose or ketose sugars? So both of these would be considered aldose sugars because the carboxyl group is located at carbon 1. All right. So carbon 4, though, is where we are basically looking at, and I'm sorry, I think I numbered that wrong on the last slide. I had that at 5. Carbon 4 is where we see the difference between glucose and galactose. And the difference is, and I know it's a little bit hard to see, if you look at this line that's kind of in the front, it's a little bit thicker. And so it's showing us that the hydroxyl group, and I'm looking at galactose right now, is pointing kind of forward, right? So if you like kind of stick your arm out, like it's pointing forward, where the hydrogen group is pointing backwards in this model. And so this, because it's pointing forward, that is making this galactose, where over here we see that the hydroxyl group is pointing backwards. It's got a fainter line and it's kind of behind this hydrogen group, right? So you can see it a little better in the space model here. So again, so when we're looking at glucose, we're going to see that that hydroxyl group is pointing backwards, where here in galactose, it's pointing forward. All right. So, and I apologize, I got this backwards. This is not carbon 1. It actually starts over here. So I'm going to fix that here if you want to fix it on your notes, just so we're correct as we move forward. Apologize for that. All right, so this is 4, 5. I was wondering if I had done that wrong because this guy here is actually 6. All right, so we'll erase that guy. And this would be 1 and 2. All right, so we were really concerned about the one at carbon 4. All right, everybody okay with this structure? Are we okay with identifying where the carboxyl group is? And are we okay with being able to see the difference? So they've colored it for us, so it was a little easy to find it here. But you can see, like, that's the only change. The only thing that's different between these two structures is where the hydroxyl group is pointing. Okay, I see some thumbs up, just so I know that we're okay. All right, thank you. I appreciate that, Bailey. All right, Amelia, Christoph, Riley. Yep, perfect. Okay, so we're good. All right. So we're going to see that there are distinct monosaccharides, so these simple sugars, and they exist because there's so many different ones, because we can just make these tiny little changes and they're going to be completely different sugars. Okay, so minor changes equal a whole new sugar. So again, you can decide where you want to put that carbonyl group. You can change the number of carbons involved. You can have different arrangements of where the hydroxyl groups are pointing in space, and you can have alternative ring forms. So there's lots of different types of sugars, right? We're not going to look at all of them in this class. But the thing to remember is that each of those sugars is going to require its own specific enzyme to break it down. Right. So those of you who've already had lab today, were you able to talk about lactose sugar? Some of you talk about that this morning in class already. Yeah. So what is lactose sugar, Christoph? Why is that one important?
It helps break down, what is it? Lactose and milk dairy products.
Yeah, so lactose is the sugar in milk, and the enzyme that breaks it down is lactase. And so if you haven't done your lab yet today, you'll get to look at that relationship and how some people, as they get older, that enzyme doesn't stay active, and therefore they can't break down that sugar and end up getting sick if they have dairy products. All right. So again, another reminder of this form and function principle. Like, my structure is different, so therefore my function might be slightly different, and it's also going to require now a different enzyme to interact with this particular molecule. All right. So again, just looking at the monosaccharides that we tend to use in biological applications, they tend to be between 3 and 7 carbon atoms. All right. So we're not talking about super gigantic, again, monosaccharides. We're looking at, fairly small ones. We're going to see that they have both hydrogen and hydroxyl groups attached to them. They tend to follow the formula that I gave you all earlier. And then you can see here, I gave some of those examples again of some of the most important ones. So glucose, by far, one of the most important sugars that we have. It's a 6-carbon sugar. It's the primary energy source in most living organisms. Fructose is also a 6-carbon sugar, and this is the sugar that is found in fruit. You can also find it put into different foods. So if you know anything about high fructose corn syrup, that wonderful invention, it contains fructose as well. And the thing with fructose is this isn't digested the same way that glucose is. Fructose is actually broken down in our liver. And so excessive amounts of fructose, especially in the form of high fructose corn syrup, can actually cause liver damage, as well as other health effects. So it's really important to understand your sugars because they're not all the same. So if you're interested in learning more about that, I have actually a whole lesson on it that I do with another class of mine. But fructose is interesting. It's okay when it's mixed in with the fruit. So if you're actually eating the apple or the peach or whatever it is, because it has fiber, and so it actually helps with the digestion of this sugar. But when we just have fructose sugar added to things, it can cause some health issues if overconsumed. Galactose, we're going to see, is a sugar that is one of the monosaccharides that's found in milk that makes up lactose. And then the other important sugars are actually found in our RNA and DNA molecules. So we're going to see these guys a lot later on in the semester. But I thought it was important to mention here, these 5-carbon sugars are going to be part of our actual DNA molecules. So they also serve a structural purpose in that regard. Whoa, it's Monday. Look at these chemical structures. What's Dr. Arrington doing to us, right? This is worse than math class, right? All right. Not really. I hope you guys get to become friends with these structures. All right. I think this is a really, really important picture. So what we see here is how glucose goes from its linear structure over here to its ring form when it's exposed to water. All right, so when you have it in an aqueous solution. We see that they've highlighted for us that lovely carbonyl group at carbon 1, and they've highlighted for us the hydroxyl group at carbon 5. Now, this is not the one that was different from galactose. This is the one I got confused, probably because of this. This is where the attachment is made. So when it forms its ring structure, we see that there's actually a chemical bond made between the oxygen on the carbonyl group here, with the carbon on this carbonyl group with the oxygen on the hydroxyl group of carbon 5. So this is where the actual ring comes and joins together. So we have a 1-5 kind of connection. All right, so where we see this oxygen in our diagram here, there's not a carbon. This one's an oxygen. So here's our 1, 2, 3, 4, 5, 6 carbons. So the 6th carbon always sticks up And we're going to see in a little bit that that's going to be important to allow for branching to occur in some of our really large carbohydrates. All right, here's another, I guess, find what's different picture, but they've colored it for us. So hopefully everyone has kind of maybe drawn their eyes to it already. So we're looking here at carbon 1. All right. And we see that, again, we kind of have this reversal of arms, if you will, right? So in carbon 1 in the top image, the hydroxide group is pointing, or the hydroxyl group is pointing forward. All right, we can see that in the bubble drawing. And in the bottom picture, that hydroxyl group is pointing backwards. All right. So the top form, we refer to this as alpha glucose. All right. And the bottom form is beta glucose. So if you want to associate beta and back, so the hydroxyl group is in the back, that makes it beta. That might help you remember this difference in structure. And again, this happens when it goes into this ring conformation. All right. The reason I bring this up is because there are big differences between whether or not we're using an alpha glucose molecule and a beta glucose molecule. So again, different enzymes are needed to break these two different forms of the glucose down. So they're going to behave differently, even though it's pretty much the same molecule, but because of this structural difference in their chemical form, they're going to behave differently within the cell. All right. You don't have to memorize all of these structures. Again, it was just so that we could see some of the differences between them. We've already kind of looked at galactose a bit. Over here is fructose. It's only a 5-carbon sugar. Well, no, 1, 2, 4, 5. No, it's still a 6, but it forms a different shape. The ribose and deoxyribose are the 5-carbon sugars. And the only difference, and they pointed it out for us, is at carbon 2. So the reason that this is called deoxy is because it's missing an oxygen. All right, so that's the difference between deoxyribose, which will be found in DNA, and ribose, which is found in RNA. So again, they're going to be very similar in structure. All right, so we looked at these monosaccharides. We see that there's a lot of similarity between them, so we have to be very careful to look at all of those little details, right? So where is my hydroxyl group? Where is my carbonyl group? Is it pointing front or back? So all of these things are things that we're going to have to be a little bit mindful of as we start building now our polysaccharides. So again, poly means many. So these are going to be our large, very complex carbohydrates. So again, things like starch and glycogen. All right, so those storage type molecules. We'll also look at some disaccharides. So again, just putting 2 monosaccharides together. You get to use your favorite chemical reaction. So those dehydration synthesis or the condensation reaction, same thing. We have a new term for a covalent bond. So this time we're going to call the covalent bonds that link our sugars, our monosaccharides together. We're going to refer to them as glycosidic linkages. Okay. And so again, it's just a big fancy name for the covalent bonds that are holding our monosaccharides together. All right. So if we look at some of the most important disaccharides that we have below, we see that I've given you guys sucrose, lactose, and maltose. So you can see here that sucrose was table sugar, and it's made up of glucose and fructose. So there's a balance between those two types of monosaccharides. Lactose, which is that milk sugar, is made up of glucose and galactose, and maltose, which is malt sugar. So if you've ever had like a malted milkshake, or I don't know if you guys are old enough yet for your malted beers, but this is the sugar in there, right? So it's glucose and glucose put together. All right. So what's the common sugar here? What popped up in all of them?
Glucose.
Glucose, right? So glucose, this is the important sugar, right? So we ingest a lot of disaccharides in our diets, but we have enzymes that are able to break all of these down so that we can work with just the individual glucose molecule. All right. So glucose is the important one. I'm sorry, Jess, I missed your chat. Which slide did you need?
Just the one before this one.
Just the one before this one here?
Yes, thank you.
Uh-huh. So
Okay, I got it.
you got it? Perfect. So we're going to see that, glucose is super important. And again, a lot of these are going to be used for short-term energy that we have. So again, we eat the sucrose and the lactose and the maltose. We're not typically going to store these, where plants may. All right, so the glycosidic linkage, again, is going to be our covalent bond that's going to hold our monosaccharides together. And this bond is going to form between a hydroxyl group on one of our monosaccharides and a hydroxyl group on an adjacent one. All right. So if we look at the structure of our monosaccharides, we can see that they all contain at least two hydroxides. But where those are located in the geometry that forms is what makes the big difference. So I think, yes, thank goodness. I was like, I need a picture. All right. So here we're going to make sucrose. All right. So that's what we're going to build here. So I have a picture of my glucose molecule. And I have a picture of my fructose molecule. And again, I'm just going to put a 1 here so that we remember that is our carbon 1. And if I look at glucose, do you think this is going to be an alpha glucose? Is this in an alpha glucose form or a beta glucose form? Does anybody have any guesses? This is a 50-50 chance, right? Anybody's like, we got the Altos question wrong. All right. Good job, Sebastian. Yes, it's alpha because it's pointing forward. So this is an alpha glucose molecule because this hydroxyl group is coming forward to us. All right. And so what we're going to see is that we're going to have our dehydration synthesis reaction take place between the hydroxyl group at carbon number 1 on our glucose molecule and carbon number 4 on our fructose molecule. All right, 1, 2, 3, 4. So we're going to remove that water molecule, right, H2O, and they're going to form basically a bond with the remaining oxygen there. Okay, so we get this kind of shape. It kind of points down. So this is our dehydration synthesis. You guys have seen this reaction several times now. And this bond that we formed here is that glycosidic linkage. All right. And we can give it a special name because I know the carbons where it was at. So I can call this a 1,4, right? I'm not going to spell this whole thing out. Glycosidic linkage. All right. So you're going to see as we start naming these bonds that we're going to add even that this is an alpha 1,4 glycosidic linkage because we know that this was an alpha glucose molecule and we've made a bond between the first and the 4th carbon and it's a glycosidic linkage. So this gives us a lot of information. We're going to practice this again. I just thought since I had some space here, we could throw that in. Does anyone have any questions? I heard someone start to say something. I didn't mean to talk over you. Anybody have a question? All right. Oops. So again, this is a structure of our glucose. We know it's aldose because my carbonyl group's at the top. I have a hydroxyl group here. So is this one an alpha glucose or a beta glucose, or can we tell? Good job, Bryson. So it's a beta. All right, so we've numbered all of our carbons. We've looked at the spatial orientation. So let's see, without making everyone go crazy, what's happening on this slide. All right, so this one, I would say if you have a little bit of time later today after class to just kind of walk back through this and kind of study it until it kind of just absorbs a little bit. So what we see here are two different forms of our glycosidic linkages forming, all right? And so on the left here is our alpha, and on the right is the beta form, basically, of these sugars. So when we look at our alpha, again, what you want to do is what determines whether or not it's an alpha or a beta is what's happening at carbon 1. So you always want to focus on carbon 1 and find that hydroxyl group. So again, if it's pointing forward, that is going to be an alpha. If it's pointing backwards, like we see over here, right, and both the galactose and the glucose, that hydroxyl group is pointing back, that's going to be the beta form. So that's the first thing you want to look at when you see, one of these sugar structures. They're always going to line up so that the carbon 1 of one monosaccharide is going to be adjacent or next to the carbon 4 of the other monosaccharide that it's going to form a bond with. So we're going to lose a water molecule, right? And we're going to basically form covalent bonds with that oxygen. All right, so we see here, because this was an alpha glucose, that we have an alpha 1,4 glycosidic linkage, right? So this name tells us a lot of information about what we're working with. It gives us a lot of information about the structure of the carbon that we're working for, all right? If we go over here on the right, we notice that we have both beta galactose and beta glucose. So they both have their hydroxyl groups pointing in the back. Now, if you notice, what if I would have had, though, beta galactose and alpha glucose, right? What would have happened there is the name would still stay the same. We would end up with a different sugar, obviously, but we're getting the alpha and the beta part of the name from the carbon 1. So if you were wondering, like, well, what if these were, one was an alpha and one was a beta? How do I know what to name it? Always name it based on the configuration here at carbon 1. Okay. So for us, this was an easy example because both of them were betas. But if they weren't, we would still call this a beta 1,4 because the beta is associated with the carbon 1. All right. Notice, though, that the hydroxyl group is pointing forward for the carbon 4, and that's going to be consistent no matter, if we're using alpha or beta monomers here. We're still going to pull out a water molecule, but notice this time we kind of have the little V pointing up, all right? So Sebastian's little signal he likes to put in our chat there is pointing up. And so this is the type, we're going to see it kind of alternates when we form the beta glycosidic linkages. All right, so you'll know it's a beta when you see it kind of pointing up, and it's an alpha when it's pointing down. All right, so that's a characteristic of how they're going to come together because of where these hydroxyl groups are in relation to each other. All right, and you're going, this is really awesome, Dr. Arrington, and I'm really glad you're showing this to me, but why the heck do I care, right? Like, this seems really detailed for biology. Everybody agree with that? Like, you're kind of sitting there going, why are we doing this? Does anybody know why I'm spending a lot of time going over this? No. So here's the thing, why this is important. We contain enzymes that can typically break down alpha-1,4 glycosidic linkages. So anything that's made with an alpha-1,4, we can typically break down and use it for energy. Things that are made with a beta-1,4 glycosidic linkage, typically we don't have enzymes for and can't break this down. All right, so we're going to see that most of our large polysaccharides that are made from beta-1,4s or beta glucose molecules are going to be used for structure, right? So like cellulose and chitin and things like that, that actually provide structure to an organism rather than provide an energy source. So that's why this is important. Lactose is a special situation, right, though, because this is found in milk. But humans are the only mammals that have maintained the enzyme past infancy to break this sugar down. And then there's some, right, as you're learning in lab this week, that don't have that enzyme. So we call it the lactase persistence gene because over time, evolution, there are groups, and this is spanning, thousands of years, where maintaining that enzyme to break down this very unusual energy source it is maintained in people's genetic code so that they're able to benefit from being able to eat dairy products and things like that. So this is an evolutionary type of gene. So that's kind of cool because most other beta glycosidic linkages that we're going to see, we don't have enzymes to break them down. So this one's very unique. All right, so here I've given you some structures of these folks. And oh my goodness, they've run out of time. So we've got starch and glycogen and cellulose. And I'll just finish this slide here real quick and I'll let you all go. So the alphas, right? We can see alphas are in all of these. So these are things that we can use to get energy from, right? There's lots of starch in potatoes and some other vegetables and things like that. We can use those. Glycogen is what we store glucose as. And so we can break that down and use it for an energy source. Cellulose, on the other hand, we can't. So what I will do is pick up with this lecture on Wednesday. So we'll start with the structure of the polysaccharides and then we'll finish that and we'll start a little bit of photosynthesis. I am going to post a pre-recorded lecture for part of chapter 10 that goes over things like what is a pigment, goes over the structure of chlorophyll. So just some really basic stuff that has to do with plants so that we can spend our time in class together talking about the actual process of photosynthesis, since that seems to be where most people get like, what the heck is going on? All right, so I'll give you guys the easy stuff to kind of, listen to at home or, if you're riding the bus or whatever, washing dishes, you can listen to me yammer on about pigments. And then you can listen to me live yammer on about how photosynthesis works on Wednesday. All right. If you have any questions about anything, just let me know. And I will send out, I'll adjust our process of science 4 assignment to next week. So giving you guys a little bit extra time to find some of those resources. All right. Because I know it just seems like everything is coming down right now. I think midterms are coming up and papers are due. So we'll give you a little bit of a breather there. And if you need anything else, please don't hesitate to reach out. All right. So I will see you all on Wednesday. I hope you have a great week. Try and get some time for yourself.
Thank you.
Yes.
Thank you.
Thank you.
Thank you.
Hey, Miss Arrington.
Yes, psych.
Hey, I didn't want you to think I left your class. Like, I literally got logged out for 30 seconds and I came back in.
Thanks for letting